Hello, everybody. I'm Greg Johnson. I'm one of Bob's PhD students, and I'm going to be talking about sort of the practical considerations behind our diffeomorphic modeling. And then, if we have enough time, I'll talk about, give you a little research showcase of some recent results that we've gotten using Cell Organizer. So I guess to hammer this home for the third time, diffeomorphic models in Cell Organizer use this measure called large deformation diffeomorphic metric mapping. And this is a morphing from one shape to another. And to address one of the questions earlier, this measure is the minimum vector field, or the minimum energy vector field, to get from one shape to another shape. So there's a whole set of transformations to move from one shape to another, but this is the one that minimizes some total deformation distance. And as Devin showed, this allows for walks through shape space that could be used to describe cellular dynamics. And it is very useful for describing spaces of cell shapes, as well as generating new cell instances. But the real question is, why would we bother doing this versus many, many other various methods, including the ones that we've shown you guys earlier? So one of the main motivations is that cells don't always satisfy the assumptions of parametric models. So this is a segmented PC12 cell that is differentiating. And using the sort of original cell organizer model, where we have a nucleus that's described as a spline, and then a cell shape that's described as a ratio between the center of the nucleus and the nuclear boundary, and then the cell boundary, you cannot get these sort of non-star polygon shapes that are typical of neurons. And there's also a bunch of other parametric shape space or semi-parametric shape space models, such as the one by Pincus and Thoreau, where they take cell shapes such as these and sample around them in a polygon shape. So each cell is parameterized by a set of coordinates that describe positions of points on a polygon. And then all those vectors that describe the cell shape are then PCA'd, and then the principal components of those are sort of displayed, such as here. So the first shape mode using this model, given these cells, is sort of the size of the cell. The second shape mode is a combination of size and eccentricity, and so on and so forth. And these describe varying amounts of different amounts of total variation of this model. And so here would be the first dimension, this would be the second dimension, and so on and so forth. But with Cell Organizer, we're really interested in modeling distributions of cells in such a way that we can generate instances from these distributions, which basically says any coordinate in the space should allow me to generate a cell that is a representative cell from the population at some known probability density. And a problem with these sort of polygon models is that if you pick points in the space, or it is possible to pick points in the space that result in degenerate shapes. So shapes where this polygon line is crosses over itself And this is, of course, not possible with cell outlines, for example. So what happens if you want to model something like this? It cannot be done. And an illuminating example of this is I used this model from Pincus and Thoreau to model the outline of neurons. And basically, we can model this distribution of outlines as a Gaussian distribution, and then sample further and further away from the center of this distribution. And towards the center, cell shapes look pretty normal. This could possibly be a segmented cell shape. But as we move further and further away, the more exotic samples from this distribution have things like the nucleus is outside of the cell, or weird parts of the cell stick out, which are not typical of any cell type. So thus motivates our use of this tool LDDMM. So this stands for Large Deformation Diffeomorphic Metric Mapping. So you're probably asking yourself, what do all these words mean? Especially, what is a diffeomorphism? So a diffeomorphism is a smooth and invertible mapping from one coordinate space to another. So here's a diffeomorphic mapping from a regular rectangular grid. So any point on this sort of screwy surface corresponds to a position on a rectangular grid of the same topology. As well, you can think of, if you look at a globe or a page from an atlas, you can find a mapping from the globe to any coordinate on a two-dimensional map. And of course, there's also a diffeomorphic mapping from someone being nonplussed to someone being very happy. And of course, this can be used to morph between nuclear shapes, for example. But this is very different from just normal linear interpolation. You cannot take, for example, two images, such as of this woman and a cheetah, and linearly interpolate them between them, because you get sort of non-anatomically correct representations. But instead, you can find a smooth mapping of all the features of the cheetah to all the features of the woman, and you can then come up with a combination of those mappings to get a sort of hybrid woman-cheetah creature, for example. And this is exactly the same methodology that we're using to find differences between cells, because we can, given appropriate images, all the cells will have, in those images, will have the same types of components, in this case, the nuclear shape and cell shape. And then we can find a minimum energy mapping from all the pixels in the cell shape in one image to all the pixels in the cell shape in another image, and so on. So an example of this is that we have this shape, shape A and shape B, which are actual real nuclear shapes. And then we can iteratively morph one shape into another that approximates this optimal minimum energy vector field. And of course, we can then integrate over that vector field to get a measure of its magnitude, which is equivalent to the distance between these two shapes. And this isn't very important, so I'll skip it. And then here's an example of a high-resolution three-dimensional cell that's morphing between this cell shape and this other cell shape via the diffeomorphic mapping between these two shapes. And as you can see, all of these, if you sort of looked at any of these cell segmentations, it's very, very difficult to determine that they are, in fact, entirely artificial. Nobody has ever seen these cells before. And of course, if you find the mapping between a whole collection of cell shapes, you can embed them into what we call a shape space, such that the distances between the shapes are proportional to the distances measured via this metric, and any coordinate in this cell shape or in this shape space corresponds to an actual cell that perhaps nobody has ever seen before, but it obeys all the topological constraints of the cells observed in this population, such as the fact that the nucleus is always inside the cell shape, and that point is always going to be sort of similar to its neighbors in that space. So diffeomorphic training, if you guys are interested in modeling your cells with this tool, we can talk about this afternoon. But the general principle of this, I guess to hammer it home again, is that we have a collection of cell shapes. We measure the distances between those cell shapes and load all of those into a matrix. And then using this tool called multidimensional scaling, we find a lower dimensionality embedding that preserves these pairwise distances in this matrix. But of course, this takes a lot of time. This method for finding this diffeomorphic distance is computationally expensive and can take anywhere from a few seconds if you have small, relatively small, like 128 by 128 by 10 pixel images, to maybe a few hours or a few days if you have very, very large images. So there's several ways. You can imagine that there's several ways to populate this distance matrix. You can, if you have a collection of cells, you can populate it in such a way that you always have a most complete distance matrix. So that would be you take one cell, or you take two cells, and you measure the distances between them. You take three cells, you measure the distances between them, and so on and so forth, and populate find a complete distance matrix for a subset of the cells in your population. And then you can embed those. Oh, there is a function that's not showing up here. Well, we can see it here. But if you have not enough time to compute all pairs of distances, you can use approximate multidimensional scaling. So if we have a distance matrix, this big Dij, and we want to find a set of coordinates that minimizes the error between this distance matrix and the distances between those two coordinates. And by adding weights here in this function that allow us to determine whether or not that distance is observed, so whether or not that we've actually measured that distance, we can find an approximate embedding of this space. And an illuminating example of why we'd want to do this is, let's say we have 250 cells, and it takes about 30 seconds to find the LDDMM distance between any pair of these cells. This would take about 10 days of computation time on a single core computer. So it may be useful to be able to find a good enough embedding given a subset of those observations. So that would be taking cells, and these would be called landmark cells, and computing the distances between them and all the other cells in your collection. So that would be populating the distance matrix like this, ignoring that, and then embedding the whole complete set of shapes in your shape samples that you have. And now that we have a shape space, how do we generate new shapes? So because between every pair of shapes in our collection, or the distances between every pair of shapes in our collection, is defined by a vector field that moves all the pixels from shape 1 to shape 2. We can propagate cells partially along this vector field to get intermediate shapes. So if I have a shape, if I desire a shape between this cell and this cell, I can move it partially along this vector field and then get a new novel shape at this point. But if I want to find a shape inside of a simplex, so inside, in between a bunch of shapes, I would then have to iteratively move between one shape and another, like this one. And then I have a new artificial shape here. Then I can generate a new shape between these 2 points, which gives me this one. And then I would be able to generate a new shape in between our second artificial shape and one of our observed shapes to get this cell shape. As well, we can model the probability density of any observation in our shape space, such that you could say whether or not this is a relatively exotic cell or a relatively common cell in some sort of quantitative manner. So there's several ways to do this. There's a nonparametric density estimation method, such that any space between cells has the same amount of density mass, such that or probability mass, such that the density in more populated regions is higher, and density in more sparse probability density in more sparse regions is lower. Additionally, we can also use the coordinates of cells in our shape space as points in any type of distribution and model populations as a Gaussian mixture model. So here is an example of a population of cells represented with black dots. And Gaussian mixture models fit on top of these with varying numbers of Gaussian distributions. And of course, this number of Gaussian distributions could be selected in a variety of manner. But this is an example that just shows how that distribution changes as we increase the number of distributions. So I guess to wrap this up, a diffeomorphic shape space is a feature representation of cells in your population, where any position in that space corresponds to a real image. And this is particularly useful because images are generally very, very high dimensionality data, as many dimensions as you have pixels. And generally, if you have a reasonably sized population, you can compress your very, very high dimensional data into a space that is only several dimensions, generally less than 5. And once you have this compressed space, you can treat this exactly like some sort of normal feature space. So now I'm going to show you some, I guess, research results that we have using this model. So one of the things that we did is we wanted to know whether or not there is some correlation between cell shape and DNA intensity. So where in the cell cycle are cell morphologies constrained, or where are they sort of restrained, such as in the beginning and the end of the cell cycle. So we took a collection of HeLa cells and computed a shape space for them, and then colored them by intensity. And here I'm showing you the first component on this plot on the x-axis, and the second dimension on the y-axis for both of these, and then the third dimension on this figure. And we can see the third shapes dimension corresponds very strongly, or relatively very strongly, with DNA intensity, which is useful for sort of, in an automated manner, determining how different components of cell morphology and biochemical compartments correlate with cell shape. You guys have seen this video several times, so I'm going to skip over it. Another task we performed was prediction of cell and nuclear shape dependency. So given a collection of cells, we computed a shape space for the cell shapes and a shape space for the corresponding nuclear shapes. And then we performed a prediction task where, given a cell shape, we wanted to know if we could predict the corresponding nuclear shape position in the shape space. And so all of these cells on the left and nuclei on the right are colored by the prediction error. So cells that are cooler colors correlate very strongly with their corresponding nuclear shapes, and vice versa. And cells that are warmer colors, such as this red, are very difficult, are a shape that is very difficult to predict the corresponding nuclear shape, and vice versa. Of course, this is very useful when trying to visualize differences in populations. Given a collection of H1299 cells that have been labeled, that have different endogenously expressed GFP labels, we wanted to know how how this endogenous expression of GFP impacts cell shape. So using nuclear and cell shape markers independent from this GFP tag, we segmented all these cells, computed a shape space, and here we're showing the first two dimensions of that. And you can see that depending on the protein that we're tagging, the modification of that protein by adding a GFP molecule drastically alters the morphology of the cell shape. And you guys, of course, have been asking a lot about modeling neurons. I also, a while ago, I took a bunch of images of PC12 cells that have been exposed to neuronal growth factor. And I was interested in how could we model the differentiation process of this. So given about 50 cells at each time point, I computed another shape space. And the first two dimensions of that space can be seen here. The blue cells being control, so undifferentiated. The orange cells, moving to warmer colors, to orange, which are differentiated cells. And we can see that the population moves from, of course, smaller, rounder, undifferentiated neurons to very large eccentric cell shapes. And of course, you can see an increased shape variation with time. And then using sort of this minimum energy deformation philosophy, we can take a population of cells at one time point and a population of cells at another time point and find a minimum energy assignment from all cells at time 1 to all cells at time 2 to say this is the most plausible precursor cell for cells at the next time point. And then this can be extended to time series of cells that instead of tracking cells over time, you're instead assigning one cell at time point 1 to a minimum energy cell at the next time point to sort of reconstruct how subpopulations may occur during the differentiation pathway. So down here is are the reconstructed pathways given a cell at a particular time point, the most probable cell morphology in the previous time point and in the next time point. As well, this deformation model can be used for not only deforming cell shape, but you can move organelles inside cells according to these deformation fields. So as the cell on the right deforms to another cell shape, we can move the pixel intensities of an observed protein distribution along with that cell, perhaps to simulate how compartments move around as cells deform. So that's a summary of some of the work that we've been doing with a very, very quick summary of some of the work that we've been doing with diffeomorphic models. Do you guys have any questions? And of course, if you guys are interested in any of applying any of these methods to your image data, we can talk about that this afternoon. Yes?
You just minimize the energy of the system?
So the deformation distance is basically the sum of all the vectors to get to deform one cell shape to another cell shape. And that deformation can happen in a variety of ways, right? So it's like if I want to move from here to the other side of the room, I can measure the distance by me going like around behind the tables and back over here. Or I can find the shortest path, which would be just walking there in a straight line. And that straight line in the shape space is defined by the minimum number of pixels that I need to, or the minimum total distance that I need to move pixels to get from one cell shape to another. Does that answer your question?
It sounds like free energy to me.
Yes,
because you're counting the ways that you can distribute this. And so it means
yeah.
it won't be the entropy of the system.
Yes, you could think of it as minimizing entropy in the deformation.
But I mean, this way of doing it, because it's segmented, right? The information about what becomes what is lost, right? So I wonder how it corresponds to the real shape change where, let's say, some postsynaptic density stays the postsynaptic density and is not free to move to other places, right?
Yeah.
So it's similar to like brain deformation and MRI when you want to assign one to another. If you did it this way, you know, the speech cortex I wonder how it corresponds to the actual growth of the cell, where things have to be the identities of certain regions have to be preserved during the deformation.
Yes. So a requirement of this method is that you have labeled similar regions in both of the shapes that you're trying to find the distance between. Or if you have a collection of shapes, similar regions, such as all the nuclei and all the cell shapes, are labeled with some identifier, which is the case in these images. I'm showing you a so all of the images in this collection of several thousand cells have a nuclear segmentation and a cell shape segmentation. But if you have cells that have sort of components that pop into existence because of some sort of gene expression, this method would not work for that.
So instead of point landmarks, there are sort of volume landmarks, right?
Yes, exactly. Yeah. So in this method, we're using landmarks that are apply to all cells and cell types. But if you wanted to measure the distances between postsynaptic positions on a bunch of neurons, you would need to have neurons that have all of those landmarks. And I think we have time, so I'm going to show results from some other results using not diffeomorphic modeling. So at this point, you guys are probably sick of the diffeomorphic model. And I'm going to talk a little bit about extensions of how we're extending Cell Organizer to model arbitrary numbers of components inside of cells. So here, I'm going to talk about what we're doing to model the relationship between structures in the cell, labeled proteins in cells, and microtubules, as well as the position of these structures with respect to nuclear shape and cell shape. So the Human Protein Atlas is a great source of fluorescence images in a variety of cell types and many thousands of proteins. And here are two typical images from the Human Protein Atlas, which all of these images have a nuclear stain, a microtubule stain, and then a stain for a particular protein in green. And the two proteins shown here are ZFPL1 and EZR. One of the sort of faults of the Human Protein Atlas is that all of these images are painstakingly manually annotated by the people who work here. And you can ask Devin about how fun that is, because he's working there and has to manually annotate these images as part of his workflow. One of the annotations, or all of these annotations, are a very small subset of biochemical compartments. So For example, one may annotate these images as Golgi or nuclear or nucleolar, but these annotations do not capture the breadth and width of the biochemical compartments available inside the cell. And so here I focused on the vesicular annotation of cells in this database. So vesicular is sort of a catch-all in the fluorescence microscopy literature for anything that is more or less punctate inside the cell body. So we identified a subset of punctate patterns, those being RNP bodies, COP2 vesicles, peroxisomes, caveolae, coated pits, and lysosomes. And these are all in the Human Protein Atlas labeled as vesicular, but they, of course, have different biochemical properties and localization patterns from just vesicles. And so what we wanted to do is, given a collection of sort of canonical punctate patterns, could we, first of all, model the localization of these patterns inside of these cells in such a way that allows us to discriminate in an automated manner one pattern from another? And then if we could do that, could we apply that knowledge to the rest of the Human Protein Atlas and perhaps take 6 canonical labels and identify any other protein expression pattern in this database as belonging to one of those labels? So you guys have seen figures similar to this this morning, where we take cell images, we model the distribution of how the components of these images are related. And then from that, we can model the distribution and generate synthetic cell images. And here I'm showing the relationships such that we generate a nuclear shape. Cell shape would be dependent on the nuclear shape. Microtubules would be dependent on both the cell and the nucleus. And then vesicular compartments, or punctate compartments, would be dependent on only the nucleus and the cell shape. And of course, if you have any knowledge of cell biology at all, you would know that the microtubules are a major highway of transportation inside the cell shape. So this independence assumption between microtubules and vesicles is not true in many cases. And so using our cell organizer model, I extended the model to allow for relationships of vesicular proteins to be dependent additionally on microtubules, as well as allow for non-punctate sort of background intensity of localization as well. I guess this is an empty slide. OK. Well. So given the collection of cells in our database, our founder protein types, we can train a model given those cells and then generate synthetic cell instances of each of those patterns. So here are 3 of our canonical patterns: coated pits, COP2 vesicles, and P bodies. And synthetic cells that are generated from the distribution learned over these example cells. And you can see that these synthetic cells are capturing variation in the distribution of these puncta that are representative of the training images. So you can see here that the synthetic coded pit cell has many, many puncta that are distributed more or less uniformly in the cytoplasm. The COP2 vesicle pattern has fewer puncta and maybe more background intensity. And then the P-bodies are relatively sparse throughout the cell shape. So this is a good, I guess, qualitative sanity check to show that our models are, in fact, learning variation of these patterns inside of the cells. Next, we wanted to know, yes?
So how would you get the covariance of the various things?
The covariance of
each of the separate cell components.
how they all covary together.
Yeah.
So, well, the thing is that the, well, we're going to get there. So we wanted to know how well our model is actually fitting the observed data. So this cell here on the bottom left, I'm showing the probability density, the learned probability density, given the cell shape, nuclear shape, and microtubules, of where we would expect to find background intensity, where we'd expect to find puncta themselves. And then if we sample from these distributions, we can then generate an artificial cell example. And looking at the fitted cumulative distribution functions for the probability of finding a vesicle or a punctate object versus as a function of distance from the microtubules versus the actual measured distribution, we can see that our model fits quite well. So what we'd expect to see on a perfect fit is a straight diagonal line. And we can see that for each of our 6 patterns, with maybe the exception of the R and P bodies, our model fits quite well. Given those models, we can therefore measure the distance of these puncta from microtubules. So I have 3 plots here, and it's our data cut several ways. So this is the average distance from microtubules for various proteins, sort of integrating across all cell types. The average position of puncta from microtubules across the different cell types, and average position of puncta from microtubules for both the proteins and the cell types. And this data is presented in a similar method to the previous figure, where we have the fitted model on the y-axis and the measured distances on the x-axis. And we can see that everything's fitting quite well along the diagonal, meaning that our model is adapting well to this variety of wide range of localization patterns. So back to the Human Protein Atlas. What we want now that we are showing that we can measure differences between these proteins and these cell types, we wanted to know whether or not we could annotate the rest of the images in the database, so several thousand more protein images, as belonging to one of our basis set of punctate patterns. So we performed a classification procedure where we decomposed our model for each cell type and protein pattern into a set of features that describe that model. And so with the exception of this feature 19 here, these are all parameters taken directly from the learned model. And because the cell types are the same across all of our images, we're going to ignore any nuclear shape, cell shape, and microtubule information. And we're just going to concern ourselves with how a particular protein localizes inside of the cell. And we performed a relatively simple balanced one nearest neighbor classification procedure with the members inside of our basis set, and from that determined an optimal threshold for similarity for these groups of cells. So it basically said how far away are images of this protein type and this protein type, and that halfway distance defines a threshold, and if you're within that threshold, you're assigned to that particular protein pattern. So we performed this classification task for our 3 cell types. And what I'm showing here is a confusion matrix. So on the y-axis is the true label for the protein type. And on the x-axis is the predicted label for the protein type, using only proteins in our basis set. And you can see that the strong diagonal, we have a strong diagonal across all of the cell types, meaning that we have a strong, given these basis set, basis set, we can distinguish between the individual patterns with a high degree of accuracy. And so what we did is that we applied this classification procedure to the rest of the human protein atlas. And here I'm showing the sort of most confident hits given members of our basis set up here, the most confident nearest neighbors or similar proteins. So gram D3 looks very similar to the COP2 vesicle pattern of Sec23V. GPC6 is qualitatively, or looking at these is qualitatively similar to FLOT1. HIP1 and DYRK1A are very similar, and so on. And looking at the literature, we were able to show that virtually all of our top hits interact directly with members of our basis set, or in these cell types, or in another cell type. And so with this, we developed a model to capture spatial relationships of punctate and background patterns with respect to microtubules. We can use this method to distinguish between otherwise very similar punctate pattern types and identify proteins previously unknown to localize in these well-characterized protein patterns. And so that's basically a survey of what we've been doing with Cell Organizer. And again, if you guys are interested in what we can do or what you can do with your data and Cell Organizer, I'd love to talk to you about that this afternoon. And any questions? Yes, Devin.
Yeah,
Yeah.
I guess my question is, across the three cell types that you looked at, how often do you see agreement versus disagreement in the subclassification?
So that is a good question. I would say about 80% of the time, that pattern shows up in at least one of the other cell types. But it should also be noted that protein localization is a condition-specific sort of response. So even though Sec23b localizes in this pattern under most conditions, you can imagine that there could be a drug condition, or if the cell's undergoing mitosis, where this pattern localizes in some other distribution. And hopefully, and we're assuming that this imaging condition is the same across all the images in the HPA dataset. But of course, knowing how hard doing actual biology is, that's probably not the case. Yes?
If you have for a protein that's on two different of the compartments that you're looking at, can you recognize that pattern and then subtract off and then identify the second compartment? Have the ability to discern?
So in this method is sort of saying that it's all or nothing, but we have we've implemented other tools to do pattern unmixing, both in the supervised sense, such as you know that there's pattern A and pattern B, and you want to identify the ratio of that mixture in other cell images, and in the unsupervised sense, such that you give a learner a learning algorithm, a collection of protein patterns, and then it learns a sort of minimal basis from which you can combine and create any of the patterns that you've observed. But that's not in this presentation. Anything else? Yes.
Yeah, so along those lines, the annotations in the APAs that we have use different quality assessments of how confident we are in the labels. And so you can have multiple main locations, or you can have sub-locations. And we also rank how confident we are in the antibody based on whether we see the localization in other cell types or tissues, or there's biochemical data to support it actually being present in that. Do you use any of that information right now? Or?
Yeah, so all of the cells have all the cells not in our basis set, we have a confidence measure on how strongly we believe that this cell type represents a particular pattern.
Oh yeah, but I meant like, so cells that you're considering from the APA, did you just take all of them and then, or did you filter them at all from the APA to get like higher confidence ones for your?
So we're using all cells and patterns that have high antibody localization confidence, or antibody tagging confidence.
So you're using some.
But those antibodies do not necessarily tag all the isoforms of the protein of interest. And so I'm doing a meta-analysis currently to determine sort of which isoform specific localization patterns we're observing. Yeah. Any other questions? Okay, I think that we're going to break until the afternoon.